everyone to our first webinar. We are pleased to share with this meeting with each of you. And we believe that arthropods are amazing creatures and we still have a lot to learn about them. Focuses on this idea, we start with this project to create a space for scientists who work with arthropods to share what they do in their labs. Each seminar consists in 2 research talks. Each speaker will deliver a 30-minute presentation with an addition of 5 minutes for questions. And there will be 20 minutes in the end for a final discussion. We hope you have the opportunity to interact with our guest speakers and ask them lots of questions. Today, we'll be learning about piercing-sucking herbivores with Dr. Jorge Zavala and Dr. Sean Prager. The first talk is titled Sting Bug Strategies to Feed on field-ground soybean and avoid plant defenses, with Dr. Jorge Zavala. Dr. Jorge Zavala is a CONICET principal researcher and a professor from the Agricultural School of University of Buenos Aires. He holds a PhD in chemical ecology and he works with plant-insect interactions in agroecosystems. His research aims at an integrated approach to soybean chemical defenses, sting bug resistance strategies, and sting bug gut microbial diversity. Well, Berenice?
Sure, sorry. Hello, everyone. I'm the other person behind Arthropodata, and thank you so much, Vane. The other speaker we have today is Dr. Sean Prager. His talk is titled Plant and Pathogen-Mediated Interactions in Sucking Insects. Dr. Sean Prager is an entomologist in the Department of Plant Sciences and Biology at the University of Saskatchewan. He holds a PhD in ecology and evolutionary biology. His research focuses on the applied and fundamental aspects of insect ecology, particularly with respect to crops, bees, and disease vectors. So now we'll be listening to Dr. Jorge Zavala first. Jorge?
Yeah, can you hear me?
Sure, yes, we can. So you should be able to share your screen now.
Okay. Can you see my presentation now?
Yes, we can. If there's anybody, sorry, Jorge. If there's anybody who's having troubles, please use the chat box and we'll try to help you.
Okay. Well, first of all, I would like to thank Berenice Romero and Vanessa Jacobi for this opportunity. I think that they are doing a great job for collaboration and know a lot of people. I think this is a great opportunity for me to show you what we're doing in my lab and also to know what other people are doing. And I hope we can get any collaboration and learn some other interesting projects. So today I want to show you some of the experiments that we perform in my lab, And the title of my presentation is Steam Bag Strategies to Feed on Field Growth Soybean and Avoid Plant Defenses. As you know, many herbivores normally feed on crops, and they feed on leaves or seeds, and they affect the yield of the crops. So, how leaf responses to herbivore damage? In general, cells can perceive the attack of herbivores by MAP kinases. These important proteins are phosphorylated and then phosphorylate the transcription factors that induce some important pathways that are related to defenses. For example, jasmonic acid, as you can see here, this is an octanol-decanol pathway, and these MAP kinases can induce the jasmonic acid pathways that induce important defenses against insects. For example, proteinase inhibitors, induction of proteinase inhibitors, or phenolic compounds like isoflavonoids. In addition, there are other important pathway that is induced, which is that produce the ethylene. Ethylene is an important hormone and is important for jasmonic acid induction because induce defenses synergistically. On the other side, when pathogens attack some leaves, other hormones like salicylic acid are induced, and this salicylic acid has important antagonist interaction with jasmonic acid. So when salicylic acid is upregulated, in general, jasmonic acid is downregulated. There are some exceptions when, for example, there are a peak of ethylene that eliminate this antagonistic relationship between salicylic acid and jasmonic acid. An experiment performed by Berenice when she was working in our lab. She used two different soybean cultivars to study the effect of defenses on from two different herbivores. One of the cultivars was more susceptible to herbivores, and the other one was like a resistant one. One of the things that she found is that the Spodoptera attack induced some defenses in the resistant cultivar, and as you can see here, trypsin proteinase inhibitors were related with the resistant one, and also benzoic acid. Well, but when he performed the experiment with thrips, she found that many other defenses were related with the resistant cultivar. As you can see here, quercetin, camphorol, benzoic acid, and trypsin proteinase inhibitors, for example. This is very interesting because it's showing that when leaves of soybeans are attacked by different herbivores, the plant can respond differentially. And also, she looked at the performance of the insects, and she found that, for example, the natural colonization of thrips was more important on the susceptible one than on the cultivar that was considered resistant. And also, when she looked at the performance of the thrips, she found that those thrips that fed on the resistant cultivar had a worse performance than those that fed on the susceptible one, suggesting that the response was very specific also. In addition, when she looked at the performance of the Spodoptera, she found that the survivorship of this larvae was not affected, and the larval mass was also not affected, suggesting that, well, the responses of the plants of these cultivars to different herbivores were different, and the response of herbivores to these defenses were different. So this is just an example of some studies that there are about leaves. How leaves responded to insect attack. But what about seeds? How seeds respond to insect attack? When we study, I mean, some years ago, when we studied to study how seeds defense against insects, there were no information about how seeds respond to insect attack. We were expecting that probably seeds respond in a similar way to leaves. So in my lab, one of the systems that we use is the southern green steam bags and the soybean. These steam bags fit on pods of soybeans and produce a very important damage. So one of the first steps that we did in order to try to understand how steam bags affect the seed of soybeans and how they respond to insect attack was studying the MAP kinases. As I mentioned before, MAP kinases are proteins that are very important because they perceive the attack and they phosphorylate a transcription factor that induce other important pathways. These experiments were carried out by Romina Giacometti. First, we decided to study the expression of the main three MAP kinases that we know that are related with defenses. And she found that the three MAP kinases, MAP kinase 3, 4, and 6, were highly expressed after herbivory. However, when she looked at the activity of these MAP kinases, she only found that MAP kinase 6 was activated. So herbivory only activated MAP kinase 6, well, similar thing with wounding, which is very important because these proteins can be expressed, but if they are not activated with mean phosphorylated, they cannot transfer this phosphate, and activate the transcription factor. One of the things that we know that we knew about MAP kinase 6 is that when MAP kinase 6 is induced, salicylic acid and jasmonic acid and ethylene are induced also. So this was performed with Lucia Barriga. What we have here is salicylic acid, accumulation of salicylic acid in seeds and jasmonic acid. Here we have after 3 hours of herbivory, 24 hours, and 72. And down here we have the jasmonic acid. We can see here that after 3 hours, salicylic acid was upregulated together with jasmonic acid. And then after 24 hours and 72 hours, salicylic acid kept up, but jasmonic acid was down, suggesting here 24 hours and 72 hours that there were antagonistic relationship between salicylic acid and jasmonic acid. However, we can see that at 3 hours, both salicylic acid and jasmonic acid were upregulated, suggesting that probably we should have a peak of ethylene. So when we look at ethylene, we found that ethylene was really upregulated after 3 hours, and explaining this lack of antagonism between jasmonic acid and salicylic acid. It was performed by Jessica Barnetto, and interestingly, after 24 hours, there were no peak, so the interaction was very clear. This ethylene emission at first was just maybe, okay, this is interesting, and maybe has a synergistic effect for jasmonic acid in inducing some defenses, but then discovered that ethylene by itself has some important role in soybean. So next step was to study how jasmonic acid is regulated in soybean seeds. We know that there are many lipoxygenases that regulate the jasmonic acid pathway. But in seeds, we know that there are 3 lipoxygenases, LOX1, 2, and 3, but nobody has shown if they are related with defenses. So Jessica start to study the expression of these loxes, you can see here the pathway. Here the MAP kinases, and here the octanose decanose pathway. Here the lipoxygenase that regulate this pathway, and this is jasmonic acid. That jasmonic acid induce defenses, and one of the main defenses and now important defenses in soybean are the PIs, proteinase inhibitors. When she studied the expression of the three LOXs, she found that only the LOX1 and LOX2 were induced after herbivory. And she didn't find anything about LOX3, suggesting that maybe these two LOXs are related with defenses. Then she also studied the expression of proteinase inhibitors, the cysteine proteinase inhibitors, which are the PIs that probably have a role against steam bugs. And she also found that after herbivory, the expression of these two genes that regulate the activity, the expression of cysteine proteinase inhibitors were upregulated. When she looked at the activity of these proteinase inhibitors, she also found that it's upregulated, suggesting that plants can perceive the attack of steam bugs, and they are able to upregulate specific defenses against steam bugs. But we are still thinking about the question, okay, how they can feed, why they can feed on soybean. So the other, there are other defenses that another important defenses in soybeans and mainly in seeds are the isoflavonoids. So what about phenolic compounds? And one of the, two of the isoflavonoids, the most important isoflavonoids in general related with defenses are diethine and genistein. These are the glycoside forms. There are also aglycons, which are like the dicein and genistein, that maybe also have a function. So since we don't have any mutants for soybeans, we decided to use another tool, which is the manipulate the ultraviolet B radiation that is well known that induce the phenolic compounds. So what we did was use different filters. These are filters that are transparent to solar UVB, which is like growing plants in the normal sun radiation, and another filters that filter specifically the UVB radiation. So what we were expecting is to have plants with normal phenolic compounds production, and other plants that the phenolic compounds were very low. Try to see the effect of these differences in phenolic compounds on steam bugs, and try to understand how these phenolic compounds, mainly isoflavonoids, are regulated. So this is one of the experiments that we performed. In this experiment, we use 4 different cultivars. And what we did, here we have the plants that were grown with low UVB radiation or without solar UVB radiation, and these ones are the plants that were grown under filters that were transparent, so with the solar UVB radiation, which means natural UVB. So we found that more steam bugs prefer to feed on plants that were grown under no UVB conditions in comparison with solar UVB radiation. Then we move we take the experiment, and when we look at the seed damage, we found that those plants that were grown without UVB radiation had more seed damage than those that grow under solar UVB radiation. So the question is, okay, what is the defenses? What are the defenses that are induced and that probably affect the behavior of these insects? So then we look first at the seeds, but we didn't find any differences between plants that were grown under solar UVB radiation and without UVB radiation in terms of isoflavonoids. Just, I mean, this in this experiment, we are just looking at the constitutive levels of isoflavonoids, not the induction. I mean, the after damage by insect. This is just the effect of UVB. So since that's why I'm no showing any data from these experiments. So we look at the pods. When we look at the pods, we found that these are flavonoids. This is lysine, genistein, lycain. These are the aglycones, and genistein. We found that those plants that were grown under solar UVB radiation, I mean, natural solar UVB radiation, had more isoflavonoids than those that were grown without UVB radiation. This is expected. And then when we look at then we made a regression analysis, and we only found significant differences in for lysine and genistein. And this is very interesting because what we found here, we have the percentage of damaged seeds, and here isoflavonoid content. And what we found is that the higher the isoflavonoid content, the lower is the damage, suggesting that these compounds have some repellent effect against steam bugs. So these compounds could be important defenses. So this is another experiment, and for this experiment, we just used two cultivars, the two most contrasting cultivars, Charata and Williams, and was performed by Francisco Dillon. And what he measured here was flavonoids, and in this experiment we used the mechanical damage and steam bugs. So now we are looking also at how these defenses are induced. When we look at the flavonoids, we didn't find any response. In seeds, we found some response. I mean, after insect damage, there is some induction of defenses, isoflavonoids mainly, but I think it's more important now for pods. So then, when he looked at the isoflavonoids in pods, here is very clear. In Charata, we can see that the induction of lysine in cultivar Charata with UVB and also genistein lysine was very clear. In the other ones, there was some induction, but no as clear in this one. Then, in order to try to understand how is the mechanism and how this expression is regulated, we decided to study the hormones related with defenses. As expected, we found some induction. I mean, after mechanical damage and herbivores, induction of jasmonic acid and jasmonic acid isolation, which is the active form of jasmonic acid. But mainly, and actually the most interesting thing here was ethylene. You can see here that ethylene was really upregulated in Charata, and this response was very similar to the isoflavonoids. So then we thought, okay, we proposed the hypothesis that ethylene regulate the isoflavonoids. So we performed an experiment. All these experiments were performed in the field, and in this case, we applied we had to control jasmonic acid, salicylic acid, or ACC, which is a precursor of ethylene. What we see here is that ethylene by itself is able to induce all the isoflavonoids. This is very clear and was very surprising for us, and we were very happy. We had some some similar results with foliage and caterpillars, showing that ACC was able also to induce isoflavonoids by itself, but this is the first evidence showing that ethylene by itself can induce some defenses. Always was considered as a hormone that can induce synergistically other defenses together, either with salicylic acid or jasmonic acid. So, okay, I show you some results about how soybean respond to Nezara Viridula attack. So we know that after herbivory, protease inhibitors are induced, and isoflavonoids are induced. So the next question was why they can tolerate seed defense. So that we proposed 3 questions or 3 hypotheses. The first one was that steam bugs in the gut have some certain proteases that are insensitive to proteinase inhibitors. Those proteinase inhibitors allow insects to feed on soybean, and they are not affected by the defenses of the seeds. The other question or hypothesis was that gut bacteria may play, gut bacteria community may, in the gut of insects, may have a role and may they activate proteinase inhibitors or may detoxify some defense compounds from the seeds. And the last one was, okay, maybe insects are able to move within the plant and avoid certain pods we are more induced, and the insects were able to try to find pods where in which the PIs or the isoflavonoids had lower levels. So what happened when the steam bugs feed on soybean? I mean, they feed on the seeds. When they suck the content of the seed, they also suck the protein. This is a protein, and in the gut of the insect, first these proteins are attacked by aminoproteases and carboxyproteases, but mainly by proteinases. These endoproteases are very important because these are the proteases that will liberate many amino acids which are essential for the insect. However, together with the protein, they also get some other peptides from the plant, which are the proteinase inhibitors. In the cases of steam bugs, the main proteases are cysteine proteases. And since soybean has many cysteine proteases, these proteases inhibitors, these inhibitors can inhibit the activity of these protease in cysteine proteases and decrease the viability of amino acids from the insects and affect the performance of the insects. So, in order to study this, Lucia Borniego and Pedro Sartoy started to study the total activity in the gut of the insect. Here we have a control. These are rehydrated seeds, and these are when the insect feed on the plant. I mean, these are in field conditions also. This is azocanelytic activity, which means the total activity in the gut of the insect. We can see here, after 72 hours, the activity of the proteolytic activity, the total activity, was decreased in the gut of insects, of steam bugs. So then we look at specifically to the main enzymes. One of the main enzymes is the catepsin L, and when we look at this is the control, which is rehydrated seeds, and also we use an rehydrated seed but boil it, and when you heat the seeds, the proteinase inhibitors are deactivated, and we were expecting to have a higher activity. However, we didn't see any change in the catepsin L, even by boiling the seeds or in those steam bugs that feed on soybean. However, when we look at the catepsin B, we found that this catepsin B activity was increased when they feed on boiled seeds, and was decreased when they feed on soybean pods. This means that catepsin B can really be in contact with these proteinase inhibitors. Is very specific, and these proteinase inhibitors can recognize the active site of these enzymes and either activate them or deactivate them, which is very important. And this suggests that the cathepsin B are more important than expected, because in general, cathepsin L is suggested as the more important in these insects. But we found the cathepsin B is very important, because just decreasing of the activity of cathepsin B decrease almost half of the, or more than half of the total proteolytic activity. This is a collaboration with Sheila Ons and Lucila Traverso, where they are working with RNA-seq and from the insect of steenbugs, and they found that 30 cathepsin, there are 30 cathepsin L and 6 cathepsin B like. This was very interesting for us, and this allow us to start to study the expression of these genes. Again, Lucia and Pedro start to study the expression of these proteases in the gut of the insects, and then they found that the cathepsin L studied was they were not affected by proteinase inhibitors. However, we found that only one gene was the expression of only one gene of the cathepsin B was inhibited, which is very surprising. We were expecting that maybe more were affected, but it seemed that maybe this enzyme is very important for, I mean, in terms of the amount of collaboration for the total activity of cathepsin B. It seems that this is a very important enzyme. So one conclusion from here, it is, I mean, these results show clearly that one of the ways that use steenbugs to feed on soybean is that they have these cathepsin L that are completely insensitive to cysteine proteinase inhibitors, and they can feed and tolerate these inhibitors. The other question, or the other hypothesis, was that gut bacterial community may affect the capacity of the steenbugs to feed on soybean. This was performed by Virginia Medina. What she did was she collected insects from secondary hosts, which means insects that were not feeding soybean, then insects from soybean, and insects from eucalyptus trees. Why from eucalyptus trees? Because these insects spend in temperate areas, like in Argentina, in winter, I mean, it's not so cold, but it's enough cold to make these insects, they spend winter as a diapause, so they hide in the eucalyptus trees. So she went and collected insects while they were on diapause in eucalyptus trees. So one of the interesting things that she found is that more than 60% of the insects didn't have any bacteria at all. But which was very interesting, and but within the bacteria that were in the insects, she were able to classify two kinds of group of bacteria. Some bacteria were transit, she called them transit microbiota, because were those kind of bacteria that were found once a while, but they never repeat among the samples. And then she found two bacteria, Jokenella and Enterococcus, that always, when there were some bacteria in the gut, these two bacteria were present. So she called them non-transit microbiota. In order to try to see if they have any function on the ability of steenbugs to feed on soybean and tolerate defenses, what she did, she performed an experiment. These are with Jokenella. These are different strains collected from different individuals. And what she did, she incubated the Jokenela together with soybean. I mean, with soybean seeds. And she used as a control the same procedure, but without Jokenela. What she found is, and I think this is very interesting, that some strains are able to really deactivate the cysteine proteinase inhibitor from the seeds, suggesting that these Jokenelas can help these steam bugs to feed on soybean. So the last question was, where do the steam bugs feed within the plant? So in order to do that, this is again Pedro, and Pedro what he did was two kind of experiment. One experiment, which is a free choice experiment, he released the steam bugs in the plant, and then he looked where they were feeding on. And then he performed another experiment, which is a no-choke experiment, in which he bagged the plants, he bagged the insect and put them on different age of pods. He divided the pods as young pods, like middle pods, starting feeding, or pods that were almost complete, and which are the more adults. So we had three different strata with the upper nodes. I mean, here the youngest, here the intermediate, and the lower, the bigger pods. So what we have here is here we have the pod damage per stratum on the y-axis, and on the x-axis we have the time of feeding. So we can see that after 30 minutes, almost 50% of the steam bugs prefer to feed on the oldest pods at the lower part of the plant, and the rest on the middle and lower part, lower stratum. But you can see that after 4 hours, these trends start to change. They don't want to feed anymore on this pod, probably because of some induction of the fences, and then after 24 hours, they stop almost feeding on these pods, and probably, they kept feeding on the younger pods. When he looked at the damage of the pods, he found, as expected, that the older pods were more damaged than the youngest pods. Then when he looked at the cysteine proteinase inhibitors of these pods, these are the pods that on the free choice. It means the steam bugs were able to move free within the plant, and these ones were and the no choice experiment. So we can see that after 24 hours, the youngest pod had lower induction than the oldest pods, and then we can see that after 72 hours, this expression decrease, and this probably they start to feed, and in these other two ages start to grow. But one interesting thing, at least for me, is that there are no changes in the free choice, suggesting that when the insect move within the plant, the induction is completely different than the ones when you oblige the insect to feed on certain part, and the plant respond very strong. So to conclude, we have the steam bugs. We can say that steam bug damage induced defenses regulated by MAP kinase 3 and 6, and modulated by both jasmonic acid, salicylic acid in developing seed of soybean. And digestive cysteine proteases of steam bugs are inhibited by seed defenses and affect their preference of feeding. In addition, genistein and dietine have repellent effect on steam bugs, and ethylene by itself induced by herbivore or solar UV radiation is enough to increase defenses of flavonoids in pods against steam bugs. This was very interesting. I mean, And then steam bugs, we can say that steam bugs avoid seed defenses by moving within the plant, by deactivating PI within the gut bacteria, and by insensitive PI proteases. We're still working on this. I mean, we don't have a complete definition, but we think and we feel that we are advanced and getting some information in order to understand how these steam bugs can really feed on soybean, even the very important defense that these plants have. I would like to thank Axel Hofer from the Max Planck Institute for Chemical Ecology. He helps a lot us with hormone identification. Sheila Ons, that we are working with the transcriptome of the steam bugs. Carlos Vallaret, that he works together for the UVB treatments, and Hugo Clotilde, that is helping us with the isoflavonoids. So here is part of the group, and we can see some steam bugs in the lab. And thank you very much for your attention. And I'm ready to answer any question. Thank you.
Thank you so much, Jorge. And please, everyone, if you have a question, just type it in the chat box, and I'll read it out loud for Jorge to answer. If typing your question takes too long, you can also raise the hand. Use that option, and I will unmute you temporarily.
Well, maybe there are no questions, huh?
Sometimes it actually takes a while to type. Oh, yeah, okay. Maria Victoria has a question, but she's typing. Great. And there's a question from Hector Cárcamo. So I will unmute Hector first. Oh, okay, yeah. So Hector, if you could wait a little bit, okay. Maria, you should be able to speak now.
Hello, everyone. Thank you very much, Dr. Zavala, for your speech. It was very interesting. Yes, my question is about the seed that you said that seeds from plants that were grown with UV radiation attenuation were damaged. I was wondering whether that damage was due to the UV radiation or after the bugs fed on those plants as well.
Your question is if there were any straight effect on insect behavior, for example, or if there are a straight effect of UVB on the plant.
Yes, if there is an effect of UVB without herbivory.
Okay, well, this is what we did here is not with lamps. I mean, we didn't increase the UVB. This is the natural solar UVB, and what we did is just remove the solar UVB radiation. If we have performed with experiment with lamps, and sometimes you can see effects straight from the UVB, but plants are adapted to the natural solar UVB. So in general, you don't see effect. Sometimes one of the effects, but it's a natural effect, is that plants are affected. I mean, growth can be affected, but we didn't see that in soybean. Maybe I saw that in other species. But you have to remember that this study was performed I mean, in natural solar UVB, we didn't increase the UVB solar radiation. This is natural. I don't know if it's okay.
Okay, so that was an interesting question from Maria, and now we have another question from Hector. Hector, you should be able to speak now.
Hello, can you hear me?
Yes.
Okay, thank you very much for your talk. Excellent talk. My question, Jorge, it has to do with the feeding behavior of the stink bugs. I'm curious to know if the juveniles don't have the ability to penetrate and damage the pods. This is something we're studying with ligos bugs in canola, and I think it's a similar interesting question. Do you find that the younger stages are unable to manage the soybean pods, and would that also interact with the defense mechanisms of the plants, depending on the stage of the pest?
I mean, let me see. It's, the communication is not so clear, so I maybe what I understand is your question is if the behavior of the steam bugs are affected by the age of the pods.
Yes, but maybe I didn't understand if you did your experiments only with adults, or did you also study the?
Oh, I see your question. All the experiments were performed with adults, and what we used was, I mean, young pods. Within the plant, you have different ages of pods. In general, the older pods are more in the bottom. In the middle, you have intermediate pods, and at the top you have younger pods. So what we did is release the insects and allow them to choose if they prefer to feed on older pods or intermediate pods or younger pods. It is clear now?
Yes, yeah, I understand that. Yeah. So I'm just wondering if you have overlapping stages of the insect, so that maybe there's some stratification that happens depending on the demographics of the insect. So with the ligos bugs, we find that the younger ones are sometimes in different parts of the plant. Do you see that with stink bugs also?
In this experiment, we use just adults. Okay. But in general, we see in steam bugs, when they are at the young stages, they feed more on leaves, and when they are too young, they feed in the whole plant. And after their restart, they already start to feed on pods.
Right. Okay, great. Thank you. That answers the question. Thanks very much.
You're welcome.
Muchas gracias.
Okay, so if there are no further questions, I would like to thank If there are no further questions, I would like to thank Jorge once again. And if there's a question that somebody might have, we will also have 20 minutes in the end. So thank you so much, Jorge. And now we'll move on to Dr. Prager, if he's there. Sean?
Thank you very much. Thank you very much for this.
You're welcome. Oh, there's Sean. Okay. Now let's. Sean, you should be able to present now.
Now I can. I couldn't unmute myself, so I couldn't tell you that I was here. Okay. You should be seeing the screen now, I think. Yes, no, maybe?
Yeah, we can see the screen.
Okay, great. Ready when you are, I suppose.
You can start. It's still.
Oh, okay. Okay, I don't know. So, yeah. So there's good news for those of you who don't want to see any more chemistry, and bad news for those of you who want to see more chemistry, which is that Jorge has done a lot of heavy lifting in chemistry. So I'm going to talk about something very related but very, very different in that perhaps the talk I'm going to give is in many ways about the larger ecological effects of these sort of interactions between plants and pathogens. So in many ways, you can think of it, this is what happens when you start having plant defenses and relationships between them. So anyway, I want to start with a really simple shape, and that's a triangle. And so for those of us who think about plant pathology or really any kind of pathology, we often think of disease triangles. And these disease triangles could be something we think about regardless of whether it's a plant or if it's an animal or a human. But almost anytime we have a disease that's caused by sort of a transmissible microbe, so something that moves around, we tend to represent it as a triangle. And of course, the most classic version of that is the one that plant pathologists like to show. And that's really the one we're going to work with to start here. And so in these classic disease triangles, you typically have a plant, which is the host and the thing that usually gets sick, a pathogen, which is some kind of microbe that is causing the plant to get sick, and the environment. And the environment in this case is really the thing through which that pathogen moves, because in classic plant pathology, of course, we're talking about rain or wind or something moving a thing around. But we're entomologists to a large extent, and so when entomologists get involved, or vector biologists, we tend to replace that environment with the vector, because the vector, the insect or the arthropod, is what's usually moving this pathogen around. And what's really important is that it's the feeding that does this. So as the insect feeds, it obviously acquires the pathogen, it inoculates new hosts with the pathogen, and it can even be a place to retain that pathogen inside it. And then all of this opens the door for a vast amount of interaction among all of these components. And it's really these interactions that our research in my lab is mostly interested in, and it's what I'm going to talk about today, using a couple of different examples. So we're going to talk about what's probably still and has been for many, many years my favorite sort of example for looking at these kinds of relationships. And it starts with plants. And in this case, the plant we're going to mostly talk about to start are tomatoes, because they're red and they taste delicious. They're also an excellent host for this pathogen, Candidata's Liberibacter solanacearum. So this pathogen, which is sometimes also called Psiloris, and they are not synonymous names, was first described in 2008, and it's a fastidious alpha proteobacteria, which is a really fancy way of saying you can't culture it in any way. It's also important in that it can survive only in its host and in a host plant, so in a vector or the host plant. But more importantly, it's the cause of many important diseases, in particular zebra chip disease, which is a disease of potatoes, and vein greening disease, which can infect tomatoes and bell peppers, and in a related form, celery and carrots and tomatillo. So this is just what it would look like if you had infected tomatoes. So here on the left, you can see sort of an affected tomato on the left, and then the stunted plant that's on the right, and then sort of the shriveling and the yellowing, and then again on potato. And in potato, we do call this zebra chip disease because if you look at that potato picture down at the bottom there, you can see that it gets sort of a zebra stripy patterning. And that resembles a zebra, at least to some people, so we call it zebra chip disease. And this disease is important because this pattern mostly comes out when you fry potatoes, which of course is the biggest use of potatoes really anywhere in the world, and it makes them unmarketable. So if you're making French fries or potato chips, you can't grow them. But it also has problems, if you look in these pictures here, where you can see that it can cause these strange aerial tubers and cupping of leaves, and in extreme cases, complete field failure. So to make this a little bit stranger and weirder, there are actually multiple haplotypes of this LOSO, of this bacteria. So there are 3 originally that are found in North America. It turns out there's actually at least 2 more that we now know of since I made this map. And then also one of these versions is invasive into New Zealand. There are also 2 more haplotypes that are only found in Europe. So there's at least 6 or 7 of these sort of haplotypes, but the 3 that are most important, or probably the 2 that are most important, are these A and Bs, and they do have a kind of an interesting geographic or regional association. So these Liberobacters are part of a slightly bigger group of Liberobacters that are very important, and they're important because they are also responsible for some other important and really damaging diseases, including citrus greening disease. So this is just sort of an idea of the worldwide distribution of some of these different species. So Asiaticus, Africanus, and American, the three on the top there, are all the ones that are responsible for citrus greening disease. Solanacerum and Europaeus are associated with other plant diseases. And then Liberobacter crescens, which I just point out for a second here with the star, is interesting because it's the only species that can be cultured. It, however, doesn't seem to actually cause any symptoms to anything, and it's found in some sort of wild tree-like species in various places in the Caribbean. All right. So again, we're building a triangle, and the triangle has three parts, and the third part is the vector, and it's probably the part that we're most interested in. So the vector in this case is Bacterosaura cockerelli, so it's a psyllid. Which means it's a hemipteran sucking insect, so it's related to aphids. It feeds specifically on the phloem, although it occasionally will get to the xylem. And it's a very efficient feeder, so it can actually access the phloem in less than 2 hours. And so we know this from EPG work. The females are very fecund. They can lay up to 1,000 eggs in their lifetime, and it takes about a month to get from egg to adult. Now, what's also important is that they feed on about 40 different host plant species in about 20 families, and we'll get back to that. So again, psyllids are an important pest, and they're an important pest both with and without this bacteria. So on their own, they can cause something called psyllid yellows, which is a problematic disease in tomatoes, among other things, and you can sort of see that here on the left where it's a yellowed plant versus the unyellowed plant. Also, they produce a lot of honeydew in their feeding, and so in some cases that will lead to sooty molds, so that alone can cause problems. They can have nutrient deficiencies if they're feeding in large enough numbers. But also, they are the only North American vector of LSO. And it's interesting because this is a change, because in the past, I would have said they're the only North American vector, but in fact, they're actually probably the primary vector of LSO. And in a few minutes, you'll understand why I say this. But in the past, really up until recently, we would have said it's the only insect that's transmitting it. It's probably not entirely the case anymore. In either case, these potato psyllids have caused tens of millions of dollars annual losses to the potato industry. And they have an incredibly large range. It essentially encompasses all of Western North America, anywhere they grow potatoes, and then also New Zealand where they're invasive. To make things even more complicated, we also know that there are multiple haplotypes of this psyllid. So when we sequence them, what we find is that there's at least 4 distinct haplotypes. Again, this map is slightly out of date. We now know there's a couple more of these too. So what we actually have are at least 4 distinct haplotypes of these psyllids, and they do seem to also be associated with certain geographical ranges. And if you're wondering, the version that's in New Zealand seems to have come from California. Okay, so having done this, we've now sort of completed our triangle. So we have our plant, our vector, and our pathogen, and we can start thinking about how these things interact. And we'll start with what arguably is the simplest version, and that is the interaction between the insect and the plant, or in this case, the insect and a few different types of plants. So one note about psyllids that makes them very important is that they're actually considered perhaps the best, outside of maybe orchid bees, example of plant-insect coevolution. So the reason for this is that psyllids, and you can see here, they're here on the left versus some other important families of sort of insect herbivores, pretty much only feed off of one genus and on a rare occasion one family of plants. So the general belief is that psyllids are pretty closely associated with their host plants. Here's another way of looking at this. So this comes out of the psyllid base, which is managed by Diana Percy and David Ullard. And what you can see if you pull out this data and make a histogram is basically that for most of the reported host plant psyllid interactions or associations, most psyllids only have one host plant for which they're associated with. But there are a few that aren't there. And the really interesting ones are that the potato psyllids and the citrus psyllids, so the 2 important insect vectors of diseases that are psyllids, are actually on this far end where they have many, many host plant species. And so that leads us to some questions about these psyllids and their host plant preferences. And the first just comes to simple things. Are they able to use all of these plants equally? And does the plant that they're growing up on influence their choice? Are all of these host plants equally suitable? And does haplotype influence choice? And so these are questions that I've been working on for a very long time, starting when I was in California and now moving actually to Saskatchewan. And so I'm going to work through some of the results that we have from thinking about these questions. Now, I'm not going to show you any techniques. The simple version for how all of this is done is simply to think of it as we have many, many colonies of insects collected from different locations off of different plants. So we have insects that were reared on tomato or potato or pepper, and that they were collected in these different locations. And then we take cages and we take a bunch of these insects. And in this case, I'm going to start by showing you only insects that do not have the bacteria. And basically, we look to see where they choose to go. So given the choice between one plant or another, where will they go? And in these experiments, what we find is that if you have these insects and you rear them on these different plants, and then you also expose them to these different plants. So for instance, you have a colony that's only been reared on tomato, and then you expose them to tomato and pepper and potato, you get something that looks like this. So this is settling behavior. So this is essentially just which plants they prefer to land on. And what we find is that they prefer tomato over pepper, which is sort of intermediate, and then potato. We can also look to see where they lay their eggs, and this is where it gets a little more interesting, because what you find is that there's actually an effect of both the rearing plant, that is the plant that you reared them on in their colony, and then also that overrides the exposure plant, but there's an interaction. So what this is telling us is that the plant that an insect starts on, that is the plant that it's reared on, influences what plant it will prefer to settle on later on in its life. We can also look to see where it lays its eggs, and here it gets sort of also interesting, because now we found a slightly different pattern. So that is, once again, the rearing plant is not actually doing anything. So there's no effect of what you grew up on, and there's no real effect of what plant you give it to, but there's an interaction between these two things and haplotype. So what we're really saying is that it turns out that if you collect these insects from California, which is one of the haplotypes, and then Texas, which is the other haplotype, there's this interaction between that and the rearing plant and how many eggs you will lay. So basically, those insects that came from pepper and were from California and Texas end up laying the same number of eggs. But there's a difference in how these two different haplotypes respond to tomato as a host to be reared on when they then go to lay eggs. Now, as I said, all of this was originally done with uninfected psyllids. We also then can go back and, still using uninfected psyllids, look at growth index. So this growth index measure is a measure of sort of how these insects develop. And here we did it looking at both, where we found to be both an effect of exposure and rearing plant. So I know this is a little complicated. I'll summarize it for you. Before we go any further, just in case you don't understand or have never seen this growth index, it's really kind of an easy way of thinking of it is that 0 means any egg that's laid never reaches an adult, and 1 means all of the eggs become adults. So basically 1 means everybody grows up really well on a plant, and 0 means nobody does. So if we look on the left side of this graph, what you see is this growth index, and what you should again be noticing is that the exposure plant, the plant to which the insects were given the choice, basically, makes a difference in some kind of complicated interaction way and relative to how well you develop. So basically, everybody on pepper develops equally well, and everybody on tomato develops equally well. Potato is somehow different. So if you are from California, which is a haplotype where they don't mostly eat potato, these insects are typically found on tomato and pepper, they actually don't develop nearly as well as when you collect them from Texas, where in fact these are considered more of a potato pest. On the right-hand side, you can see the effect of the rearing plant, which is sort of a different effect because basically what's happening here is that we're finding that, is that the colony again has a little bit of effect on how you develop. So if you were from a colony that was mostly fed potato, it turns out you develop a little bit better. All right. Sorry, I'm having a little bit of technical issues. if we summarize this, just to make this all easy for you, what it really comes down to is if you're from California and you get pepper, you're pretty happy. If you're from California, you get potato, you're actually not so happy. If you're from Texas and you get a potato, you're pretty happy, and everybody likes tomato. Now, having said that, again, all of this was done with uninfected insects. But it turns out that infection can affect behavior. So the presence of pathogens in vectors is known to change the behavior of the vector. And similarly, the presence of pathogens in plants is known to change the behavior of those plants, or rather of those vectors when feeding on those plants. And here's just a bunch of examples of where that has been found. And we also know that to be the case for these potato psyllids. And in fact, what happens is that if the potato psyllid is infected with this Liberibacter, they preferentially go to uninfected plants. And when the insects are not infected with Liberibacters, they preferentially go to infected plants. And the presumption here is that this is the pathogen manipulating the insect in a way that promotes its own spread. So because of that, we went back and did these experiments again. And in this case, what we did is we looked at the effect of being infected or uninfected on these same sorts of choice tests. And here what we found is that only on pepper and only to a very minor case does infection make a difference. So the infection is not changing where these insects prefer to go. But it does change, at least in potato, whether or not they lay eggs. So there's some effect of being infected on the number of eggs that these insects either can lay or choose to lay, which suggests that the bacteria in some way is not just changing behavior, but it's changing it only relative to certain plants and to certain types of interactions. Okay. That's the easy version. But the truth is, if we start thinking about things like crop fields, what you realize, why is this doing this? Sorry. Is that they're really a mosaic of insects and microbes. That is, a field isn't just one set of plants and one insect. It's actually many plants, if you include things like weeds, and many insects and many microbes, and they're all interacting. And so often, your disease triangle looks something like this, where there's multiple, even more than 2 pathogens interacting with that plant that's part of this disease triangle. And we can model that too, and in fact we have. And we did that by taking the same system with our Liberibacter and psyllids and tomatoes and adding in tobacco mosaic virus. So tobacco mosaic virus is the most well-studied virus in the world, and it is a mechanically transmitted virus that infects all kinds of solanaceous plants, and that includes tomato. So if you do this, if you take these tomato plants and you infect them with tobacco mosaic virus, and then you give the psyllids the choice of infected or uninfected plants, so similar idea, you imagine a cage where on one side there's infected psyllids and one side there's not, what you see is something like this. And what this is that there's always a preference in the psyllids for the uninfected plant. That is, the insects like the healthy plant more than the infected plant. And they like it better for laying eggs as well. So if you do this, they will lay more eggs on the uninfected plants than the infected plants. And the same thing happens if you do it in a choice or a no choice assay. So even if you just cage these insects on and don't give them the choice, that if you just take either an infected plant or an uninfected plant and you put the insects on, this holds. So they still don't like it, even if they don't have, regardless if they have choices or not. But then if you take those eggs in the no choice assays and you let them develop, what you find is there's not really any difference. So it's not a preference that is based on the ability of these insects to develop, it turns out. It's just something that they don't like, the sick plants. But the important part about this is probably that it alters the transmission of Liberibacter. So now what will happen is, if you're thinking about this, that these insects have this Liberibacter, and then you go and look and see how many of these plants then were able to be infected. So you have an uninfected plant and an infected plant. One is infected with TMV and one is uninfected. And then you put in insects that you know have the Liberibacter, so that both pathogens are here. And you go back and you do some qPCR to see if they're infected. What you find is that the proportion of plants that are infected is higher in healthy plants, those that don't have TMV, than those that do. And this also translates to titer. So this is just raw CT value for anyone that's not used to seeing this. Basically, a lower value actually means more bacteria. And what you see is that uninfected plants, plants that do not have TMV, are actually more likely to have infection with Liberibacter, and they are more likely to have a higher titer. Okay, now we're getting really crazy. Because the truth is, not only can you have something that's mechanically transmitted like TMV, but in fact, these triangles can overlap, because it's not unreasonable to have multiple pathosystems that are at the same time. So you can have more than one insect in the same field that's transmitting more than one pathogen. And it turns out we can model that too. So this is mostly work that's done originally in my lab, and then a lot of it now done by Dr. Regina Cruzado at the University of Idaho. And so what we did is we took two pathosystems. So we took the original one we were working with tomatoes and the Liberibacter and the potato psyllid, and then we also added in green peach aphids, which are a vector of potato virus Y, which is probably the most damaging vectored pathogen of potatoes, and probably the second most damaging pathogen of potatoes. So it's very common for these things to occur at the same time. So it's very reasonable in both tomato and potato to have both Liberibacter and potato psyllid and peach aphid and potato virus Y all in fields at the same time, or at least at some time during a season. So this is a bit of a real world scenario, but it's one that allows us to do some experiments. So we did the same sorts of experiments as we did before, except now we mechanically inoculated the plants with potato virus Y instead of inoculating them with TMV. And this is a similar kind of result. So once again, what you can see is we're looking at the preference. It's shown differently, but it's the same idea. So basically, gray bar means preference for PVY infected plants, and blue means PVY uninfected plants. And here I'm showing the positive and negative psyllids on the same graph. So positive and negative, again, meaning the psyllid itself has the pathogen. And what you can see is that for the most part, there is no preference. The exception is only in the 24-hour time point, and only when the psyllids are LSO negative. So in most cases, what it suggests is that where the TMV infection changes their settling behavior, this doesn't seem to be the case, or at least not very much, in the PVY infected plants. Similarly, you can go back and you can look at oviposition. So this is the number of eggs. Again, same idea. And again, there's no difference in the negative plants, but there is a difference in the positive plants. So PVY negative plants are subject to more oviposition. So more eggs are laid on the negative plants than on the positive plants. And then finally, in the same way, you can look and see if PVY infection in the plants influences infection with the Liberibacter, and the answer is no. So neither the titer nor the incidence of LSO differs. So what this says is that where the TMV seems to make a distinct difference in the insect's behavior, and that behavior translates to infection of the plant, that's not the case in PVY, which is a vectored pathogen. Okay, you confused yet? I hope you are. I'm trying to get you all confused. Because really all I want you to take away from all that is a simple thought, which is that plants can mediate interactions in all different ways, and that includes between pathosystems. Now, like I said, I saved you the chemistry in this case, but the truth is we have some ideas of why some of this is happening, and it has to do with both how the different defense pathways are responding to these different types of infections, and then also how the important secondary and primary metabolites affect it. So it's just worth realizing that what's really happening in many of these cases is that the plant is being changed in ways that the insects are responding to. Okay. It's also worth knowing that pathogens can have multiple vectors. So not always is it the case that a pathogen, a single virus or a single bacteria, has only one vector. And of course, pathogens can also have multiple hosts. So just like insects can feed on multiple hosts, so can pathogens. And turns out we can model this too. And in order to look at this, we have a very convenient scenario that we've come into. So in Western Canada, where we are, we have a bit of a problem in our ash trees. And the problem is that they're dying. And this ash tree death seems to be associated with another type of psyllid called cottony ash psyllid, or at least that was what the reports are. So these are also small phytophagous hemipterans. They're in a different phloem feeding superfamily than the potato psyllids, but they're closely related still. And they are a specialist on several species of fraction. So they're ash specialists. And they're endemic to Europe with an incredibly large distribution that does include North America almost exclusively as an invasive pest. And there are occasional records of this insect in North America going back well over 100 years, but they seem to rarely establish or become common. However, in around 2000 and since then, we've been finding this insect a lot in various cities in Western Canada, and that includes Edmonton and Calgary, Alberta, and now Saskatchewan. And they're usually on black ash or Mancana ash or hybrids of those two. And we often do both hybrids and grafting for various reasons onto green ash. And it turns out that anytime people found these ash trees dying, they seem to find these psyllids. So this is just sort of what it looks like when you find these ash trees that are sick and associated with the psyllids. So you get a couple of things. The first is you get this sort of cupping and this pseudo-galling. And this is a somewhat common symptom of psyllid feeding on trees in general, but it's a particularly bad one in this case. Here in the middle, you can actually see the insect on the plant. And then on the far right, you can see the complete loss of leaf canopy and eventual death that is associated with these insects. However, we're a little curious of whether or not these insects are doing damage all on their own. And one of the reasons for that is that this kind of damage is not reported in Europe where these insects are endemic. So we were curious if maybe there was a little more going on. And to look at that, we started by collecting these insects. So we put yellow sticky cards out into trees, and these are sticky cards designed for citrus psyllid, but they work pretty well on almost all psyllids. And then we also directly collected insects off of trees. After we did that, we did PCR using these universal primers that we already had for psyllids, and then we Sanger sequenced it. And then from that, we did two things. The first is we developed some qPCR specific Liberibacter primers for these particular insects. But I'm getting ahead of us. We also did some sequencing and blasting and bold searching, so barcode of life searching, against samples we had. So from that, all that sort of DNA work, what we got is this. The first is that we found that basically, as we expected, the insects that we were collecting are in fact cottony ash psyllid, and they do match almost exactly to both two different genes, both taken from a type specimen that was collected in the cottony ash psyllid's natural range in Europe. And that sample came from Essex in the UK. So first thing we confirmed, definitely cottony ash psyllid. But then the other thing we did is we took that DNA that we extracted and we also used our degenerate PCR primers for Liberibacters to amplify about a 1200 base pair region. And from that, we could then go and look and see what kind of Liberibacter we had, which we obviously knew we now had. And then we also made qPCR probes specifically so we can actually use those to now detect and do experiments and confirm whether or not future insects are infected. Something interesting happens, though. If you take the Liberibacter we sequenced and you blast it, this is where you find it. So it falls out right in the middle of the Liberibacter, the Solanacerum, the one that's causing zebra chip disease, right in the middle, bell pepper and potato and tomato from the United States. So basically what this tells us is that we were finding infected psyllids with Liberibacter, but not a different Liberibacter. We were actually finding the same Liberibacter that was causing potato psyllid related diseases. So we thought a little farther about that, and we went a little bit deeper into our analysis, and I don't have a good picture of that. But what it actually turns out is we have one of these weird Solanacerum haplotypes. So when I said it's one of, not the only anymore, that's because it turns out that there are a couple of other haplotypes of this Liberibacter, and they're associated with a different psyllid, and that different psyllid has a different host, but they all seem to be very closely related Liberibacters. So all of that tells us something kind of interesting, and it leads us to this story that we've been telling, although not yet confirmed. So what we think happens is that our potato psyllid is sometimes feeding on field bindweed, and this has been reported multiple times, where field bindweed, which is this sort of weedy plant in the Pacific Northwest is occasionally a host for this psyllid, for this potato psyllid, and occasionally can test positive for Liberibacter, although it seems to be a different version of the Liberibacter. But it also turns out that there's a different psyllid species that also carries this different version of Liberibacter that also likes to feed on field bindweed. So now you have two different psyllids feeding on field bindweed, and the field bindweed having Liberibacter in it. And so really what you have is the field bindweed sort of serving as kind of the host for two different slightly related pathosystems where both insects are feeding on it and both transmitting the Liberibacter to it. But what we think may have happened is a bit of a shift, perhaps to the right, where the bindweed eventually got replaced by the ash tree. So what we think happened is you have this happy relationship that's been going on probably for quite a while between these two psyllids and the bindweed. And then we added in this ash psyllid, which also was feeding on the bindweed, perhaps because bindweed is a generally attractive host for all psyllids. It got infected with Liberibacter, moved back to doing what it's supposed to do, which is feed on ash tree. And now we have all of these infected ash trees in the western part of Canada and part of the United States as well. Okay, so these aren't necessarily questions that have to be asked with psyllids and Liberibacters. We can also ask them with other pathosystems, and that's exactly what Bernice in particular has been doing. So she has a slightly different but actually quite related pathosystem, and this one uses phytoplasmas, another type of sucking insect, the aster leafhopper, and a whole different set of host plants, including various asters and canola and cereals. So just to put this in perspective, at the top here, you can see where LSO falls out. These are the alpha proteobacteria at the top. So these are the Liberibacters. And then this blue box, these are the phytoplasmas. And these are all sort of related molecule-like plant pathogens. So we're still talking about primarily fastidious bacteria. So most of these can't be cultured, and most of them are associated with one kind of disease or another. And most of them are usually associated with either plants or animals as their primary host. Similarly, the vector that we're talking about is fairly closely related. So here are psyllids falling out down here along with the aphids. And they are basically a sister group to the group that contains plant hoppers, and then the leaf hoppers, which is the group here. And of course, these are all sort of sucking insects. They all have piercing sucking mouth parts, and are all true bugs. So we can again start with the same kinds of questions. Bernice is interested in these same kinds of questions, exactly like we have been thinking about with these potato psyllid systems. And that starts with which of these plants that are reported to be hosts for the leafhopper are hosts for either the leafhopper or the pathogen. And again, she can ask it using both infected and uninfected insects. And again, she can ask it by exposing these different plant species to these insects. And so one of the things that we can ask, just like we did previously, is what is the effect of AY infection on host plant suitability? So same idea, if the insect is infected or not infected, or if the plant is infected or not infected, will it change their preference to these plants? And this is important because understanding these things, while they're also ecologically interesting, is also going to help us figure out why some plants that are grown in the Canadian prairies get really bad infections with Aspergillus phytoplasma while others don't. And so Bernice sort of did the same thing. She took a whole bunch of plants. So canola, which is the one that is most commonly thought of as a plant damaged by Aspergillus, but then also cereals, where we also know we can find the insects and the pathogens, and then a bunch of weedy plants, and then also Arabidopsis. And Arabidopsis is here not so much because it's naturally occurring in the prairies, but because it opens the door to do all kinds of other experiments where we can look at sort of the chemistry and gene expression and plant defense pathways that are regulating some of these patterns we find. So in a similar manner to previous, Bernice can do these no-choice bioassays. So she takes 3 pairs of the insects, cages them onto the plants, and then looks to see what happens. And she once again calculates growth index, just like we did earlier. And she can basically look to see how many eggs are laid on a plant and then how they develop. And what she finds is this. So this is pretty complicated. I'll try to work you through it. First thing you need to know is all the white bars represent leafhoppers that did not have the aster yellows, did not have the phytoplasma, the bacteria, and all the gray bars did. So if she looks at the number of offspring, so we don't actually count eggs because it's destructive to do that. So she actually counts first generation nymphs. So first things are nymphs. It's a very good proxy. This is what we find. Arabidopsis not only has a relatively moderate number of offspring, but it's strongly influenced by whether or not the insects are infected or not. So the uninfected insects lay more eggs on Arabidopsis than the infected ones do. Barley is quite a good host for laying eggs, but it doesn't make a difference if you're infected. Here's fleabane. So here's the slightly different pattern, but it's not particularly significant. Here's oat. Here's wheat. So on all of these plants, while there is a little bit of effect, the important part is that they are pretty good for laying eggs. The insects like to lay eggs on them. Here's canola. So one of the things you should be noticing is the plant that we worry most about as a plant that's diseased by this phytoplasma is not one that the insects like to lay eggs on. Same thing with sow thistle. Dandelion is sort of in the middle. This is interesting because these are aster leafhoppers and these are dandelions, so they're an aster. Here's marigold. Again, kind of marginal. So just like in the past instance, Bernice went in and she calculated this growth index, and you can think of it exactly the same way. So once again, if you're at the top of this graph, if you're a 1, it means the plant is very good for insects to develop on, and a 0 means they can't develop on it at all. So here, Arabidopsis is quite good. Barley is quite good. Dandelion is quite good, although it varies quite a bit between whether or not you're an infected or uninfected insect. Fleabane, marigold is highly variable. Oat is quite good. And wheat is quite good. On the other hand, canola, not so good. And same thing with sow thistle. So what this is telling us, and this is why this is really important, is that the plant that we worry most about relative to disease is actually not a very good plant for these insects to develop on. And that's particularly weird because it suggests there's something different about becoming infected or showing symptoms and being used as an actual host plant for these insects. So this is stuff that Bernice is still working on, but she was curious if it somehow reflects some sort of preference in these insects. So she actually did choice assays and went and looked to see where these insects prefer to go. And what you can see is that there's clear preferences, and usually it's for a crop. So in most instances, it's not actually the wild, sort of the weedy plants that these insects like, it's actually the crop. And that even includes when canola is put against some different weeds. So one possibility for some of these things that we're finding is that crops are perhaps less capable of fighting off these insects, so their defenses are maybe not quite as good as the natural crops, and therefore when the insects have a choice between a weedy plant and a more developed plant, they're going to go to the more bred plant because it doesn't defend itself as well. So finally, just to make this extra complicated, Bernice also goes and dissects all of her insects. And by dissecting them, it allows her to look at the number of eggs that the insect has. So these insects are not born with their full egg complement. That means that as they feed, they are developing eggs. So the quality of the plant on which they feed for nutrition theoretically should influence the number of eggs they have. So a better plant should lead to the insect having more eggs that she can lay, but then also more eggs when they are dissected. And again, Bernice can do this with both infected and uninfected insects. So she can see whether or not the pathogen is putting some kind of burden on the insect that leads to her having more or less eggs. And when you do that, you get this. So what you can see is that for the most part, there isn't a lot of difference by carrying these pathogens with you, but there's clearly a difference between, or I should say among, the different plants with respect to the number of eggs that they can develop. So some of these plants are better for these insects to develop eggs than others. For example, dandelion is very bad. They don't seem to be able to develop many eggs when they're on dandelion versus barley. And notably, barley is actually the default plant we use in our colonies, where they actually are pretty good at developing eggs on barley. And if you remember back, these are also a pretty good correlation between the plants on which they lay eggs and the plants on which they have these higher egg loads seem to be related. So one possible thing that we're starting to wonder about, that this leads us to think about, is that perhaps you lay more eggs on the plants on which you have more eggs, and you may have more eggs because they're better plants for you to develop eggs on. All right, so to put all this together, really what the whole point of this talk, the whole point of what I'm showing you everyone is to take away is that we often think of these things as being very simple. We often think of an association between, say, an insect and a plant, or a pathogen and a plant, or even sometimes an insect and a pathogen and a plant. But it's not true. These are incredibly complex interactions in many cases. They're multi-trophic, they're influencing each other, and they're usually mediated by the plant. So all of the things that a plant does in response to attack and its ability to defend itself against everything that's coming at it is going to affect both the way the pathogen does things and the way the insect does things, and then all of the dynamics that surround that. And this is really important as we become better and better thinking about how we should manage plants, both for crop yield and for breeding, and then for just sort of disease management and insect management. It's important to realize that this is the case. But then it's also really interesting for ecological reasons, because what it suggests to us is there may be a lot of things that are driving both ecological relationships and perhaps evolutionary ecology relationships, how these things have evolved together and why different plants produce different compounds. And all of these things might be a bit more sophisticated than we tend to think of, because often we're only really thinking of it in the context of the individual pairings that we're most interested in. All right. So there's a tremendous amount of data I showed here, and there's many more pieces of it if we were to keep going. So it was paid for by lots of different places, and I want to just make sure everyone gets recognition of all the different people that paid for it. And then, of course, all the different people that did the work and helped do the work and helped pay for the work and are in every way associated with it. So everyone in my lab at Riverside before I moved here who did some of this, and our collaborators in Idaho and at Cornell and at Canada. And with that, I will take any questions.
Okay, so thank you so much, Sean. Now, if anybody has a question, just let me know and I will unmute you, or you can also write them using the chat box. Abigail, you should be able to speak now.
Okay, thank you. Hi, Sean.
Hi, Abby.
So I was wondering if you thought that, you mentioned that basically when you were looking at LSO versus potato mosaic virus or tobacco mosaic virus, they didn't really seem to prefer plants that were infected with that. There wasn't the same effect when you used another bacterial pathogen, which was that was something else. Do you think the difference between those two was a viral versus a bacterial difference as well? That one bacteria is able to maybe share a plant better with another bacteria compared to a virus?
Okay, so it's two viruses. So it's the differences are potato virus Y and then tobacco mosaic virus. But that said, this is where all the chemistry I didn't show comes in. It probably has to do with the actual differences in what happens when you're infected with these two different pathogens. So tobacco mosaic virus, if you were to go in and look at the chemistry that I didn't show everyone, has a couple of really important effects on the plant. And one of them, it changes the concentrations of sugars. So you get more happy sugar and less unhappy sugar, let's say, in tobacco mosaic virus infected plants. So there's less sugars in the TMV infected plants. So presumably they are less nutritious versus the uninfected plants. So uninfected plants, let me back up. Uninfected plants have more sugars than infected plants. There's also a bunch of differences in amino acids, including phenolics and terpenoids. So all the compounds that we expect to either be different in their nutritional quality or in being associated with plant defense, including eucalyptol, which we know is a plant defensive associated compound, are higher in the uninfected plants. So basically, TMV seems to change plant chemistry is the short answer in a lot of different ways. And when we look at that in PVY, we don't find really any of those same kinds of differences. And so that's probably what's actually happening. But whether or not that's the difference of the virus or because one is mechanically inoculated and one is vectored are all the questions that remain to be answered. The other thing, and I didn't get into this is another chemistry thing, is that we also know that there are differences in osmotic acid and salicylic acid and ethylene that are associated with these different infections.
Okay, and now, Sean, we have a question from Patricia Fernandez. She asks, do you have any idea of the cues that insects are using to find the plants? Differences in plant volatiles could explain differences in plant preference, maybe?
We don't exactly know. So we certainly know that volatiles make a difference in some of these relationships, but we don't know about the volatiles specifically in the PVY context or in the TMV context. So we have not gotten so far as to collect volatiles in other cases. We do know that if you look at the preferences related to the PVY, to the Libirobacter infections in these two different plants, then there are clear volatile differences.
Okay, and well, Patricia says, thanks a lot, great talk. And then there's a question from Dan that says, you mentioned that with Bernice's work that the leafhopper colony was raised on barley. Do you think that that may be contributing to their preference or ability to utilize cereals?
Well, Bernice might be better capable of answering this question than me. The answer is probably not. So while I do think that, like we saw in the potato salad, there may be some sorts of colony plant effect, it seems unlikely that that's the biggest thing going on here because what she's finding actually kind of tracks with what we see in the field relative to where you find these insects.
And if I can add something to that, we did try to have another colony on wheat, and I did try another colony on fleabane, which is not a cereal, but it was like one of the best non-crop species for in terms of like oviposition. And both colonies were like, I wasn't able to actually raise them on those other plants. So far, it's actually barley, the one that helps me keep the colony going. Maria Victoria Gol Arauz, thanks to you for your talk. And she says that she will be in contact because she's working with a similar pathosystem with maize, Dalbolus mites, and corn stand spiroplasma. So, cool.
Oh, very cool. That's very cool. We don't have a spiroplasma version of this, so that's perfect.